Morning, everyone. So thanks for the introduction, Paul. So I'm Ben Douglas. I'm the field application specialist for the Salentum portfolio based in the East Coast. So I cover, all of the legacy instruments. So our VIPS Pro, our Cellmetric X, our Icon, and all the associated software with it. And my talk today is going to be mainly focusing on how we develop neural networks to accompany these instruments on the software. And I promise I will not dive into any of the hard maths that involved with creating these neural networks. I know it's super early and the caffeine isn't kicked in yet. So I promise I'll just give the high level overviews and not dive too much into the weeds. So I'm just going to start off with acknowledging the team that was involved with this project. I unfortunately was not part of the dry lab team that developed these neural networks. I'm field-based. The folks that did do this are much smarter than I am. And we'll dive into some of the work that they did that really make these products stand out and shine. Then we'll just do a brief overview of artificial intelligence and how it applies to our workflows, how these are created, and then some example and use cases of how these are showcased in our products. And then also touch on a new neural network that we just launched with our CMX called Automated Evidence of Clonally. So first off, I know there's a slew of folks that I would like to thank, specifically the wet lab and the dry lab teams out in the UK, primarily Anna Davidson's team and Camilla's team, as well as the leadership team under Neil Pomeroy, Andrea Goff, and the product managers, Paul Butler and Martin Rucka. Without these folks, we wouldn't have any of the neural networks that are currently in our products today. So to kind of kick things off, very oversimplification of what artificial intelligence is. And at its purest form, it's a field which combines computer science and robust data sets to enable problem solving, encompassing both machine learning and deep learning, as well as algorithms that are used to create expert systems which make predictions and classifications based on input data. So as I said earlier, all three of our instruments currently utilize a neural network in some capacity. So our VIPS utilizes one to classify cells within the droplet after it's deposited to a 96-well plate, a 34-well plate, thus creating the first of our double lock of assurance. The second one that was implemented was our trypan blue exclusion neural network, which is utilized in our Icon for the viable cell density assay. And then the newest one, like I mentioned earlier, is our automated evidence of clonality, which is launched with our Cellmetric X. And then, so just real briefly, so this is essentially the general outline that our team in the UK utilizes in order to create these networks from the ground up. So it consists of four main chunks of work, essentially. So data prep, network dev, network training and testing, and network validation. And so I'll dive into each one of these again at the highest level possible without diving too far down into the weeds and the mud. All right. So the first piece is the data prep, which is pretty much is where all the heavy lifting is involved with the wet and the dry labs out in Wimborne. So we have our scientists meticulously pipetting out the cells, and ideally, we try to capture as many images and as much data as possible. There's a lot of different permutations that can exist when we're seeding out these cells. Obviously, it would be nice if all of the wells that we pipetted out were clonal. Would be ideal, but I think everyone knows that's not necessarily the case here. Then those plates are then fed into the instrument of choice. In this case, I'm going to be diving mainly on the Cellmetric X. And those images are captured and then manually labeled by the dry lab scientists, kind of indicated by this last column here. So we try and capture as much data as possible. So the single cells that are in suspension, the single cells that are adherent, the artifacts in the plates, the imperfections with media debris, clusters, multiple cells that are present, doublets, as much data as possible so we can feed it into the neural network and then train it to its ideal state. Right. And then, so next piece is the development and the training section. So here we have a very rough schematic of a very basic neural network. This isn't the exact one that's in our instruments, but it'll serve the purpose of kind of describing the overall flow of it. So each one consists of a input layer, which is indicative from this far left column here. And then this is where those images that have those, essentially all those permutations that I was chatting about earlier. So single cells, adherence, cell size, morphology, shape, color, all those are sent into the network and the input layer. And then they pass through a series of hidden layers, which are these two columns here. And then all of these interconnecting lines here are different weights and biases that essentially are utilized in order to train the network and then ultimately put out a specific output, which is indicative of these two columns here. So in this instance here, we're showing an example of an undertrained network. So we see here that ideally we want to classify all of the cells that are present in the well here. What you can see here, and I know it's tough because they're a little tough to notice from afar, but some were missed. We missed a cluster here right in the center and then a few cells around the periphery, which ultimately not what we want. So we would ultimately adjust the weights and biases within the network in order to create a more optimized network before we launch it into the wild, so to speak. And then here's essentially just another slide stating exactly that. So we have a bunch of training data that's being sent through the network, which is the stack here. It's passed through again, and then it produces what it thinks is the correct classification. Ultimately, sometimes it misses, and then the error and the loss is calculated, and then it's fed back through the network via backpropagation in order to create those minute tweaks that will ultimately lead to the correct classification down the road. I just want to pause here real quick. Are there any questions? I know there's a lot of information being thrown out at everyone all at once. No? Okay, fair enough. And then so the next bit would be obviously once the training is deemed sufficient, the dry lab scientists will then implement an early stopping to prevent the the network from becoming overfit, and then ultimately creating a more validation error and training error. So ideally the points that they want to stop the training is right here called early stopping, right before this inflection point where it ultimately we see an increase in validation error as a result of overseeing the same data over and over again before it's passed over to the validation sets. And then ultimately, so once the network has seen all of the training sets of data, it's then fed a set of new data that it hasn't seen before, which is the validation sets. And then if it correctly classifies all of this, all the data that's fed into that set up into a certain percentage, then it's then fed back to the folks at R&D to further validate it before it's released into the final version of the software. All right. And then, so as I alluded to earlier, we have a few different flavors of our neural networks. Each one has a very specific function. The first being the VIPS Pro. So it utilizes a neural network to detect the cell within that droplet after it's deposited. Again, creating this first of the two blocks of assurance for clonality. I think most of the folks in the lab here and then online are familiar with the VIPS. Detailed explanation later on during the day. And then the second one is our ICON, which utilizes the neural network that's used to classify live and dead cells via trypan blue exclusion. So it's a little bit specific purpose of downstream of the VIPS and the cell metric. But it's utilized mainly to, again, classify live dead cells at the shake phase and part of the scale-up process in order to further rank your clones. And then, so I briefly wanted to talk about the newest neural network that we've implemented in the CellMetric X, which is our automated evidence of clonality network. And to do that, I wanted to set the stage and kind of talk about how clonality assurance is done currently using our legacy instrument, the CellMetric X. And I would imagine most of, if not all of you guys, probably employ a similar workflow. So you lay your cells down, let them grow out to day 14. You get a nice colony and then track everything back through the subsequent scan days to day 0 and then prove the assurance of clonality that way. Is that hitting home for most folks? Is that, yeah, okay. Most of the time? Okay. And then ultimately, at the end of the day, we were able to add annotations to the specific that specific cell in the well, and again, create our assurance of clonality. And then this typically, depending how experienced the user is, this varies between about 2 to 4 minutes per well. Is that kind of the rough approximation as you guys are currently seeing? Yeah. It'd be nice if it was a little bit faster per well. So to track back from day 14 and go through all those subsequent steps to day 0, and then label the cell in the well, roughly. Sorry, a minute? Okay, less than a minute. Yeah, that's fair. So with automated evidence of clonality, the experience is much faster. So instead of having to manually find that cell in that well, the software will do it for you. So on day 0, you're able to essentially find that assurance of clonality much faster and use the AEC algorithm as a tool to further confirm your judgment of whether or not that the well is in fact clonal or not. So we're able to actually do this in a fraction of the time. As just mentioned earlier, on average, again, depending on how experienced the user is, it takes about 2 to 4 minutes to find that cell in the well. Whereas here, we can do it in about 30 seconds on day 0. And then also the algorithm, again, it works very similar to the way that the VIPS works, but instead of classifying the cells in the droplet, it's actually doing it at the whole well level, which is ultimately the end result. In combination with the VIPS droplet, this provides a really nice, assurance of data when you're submitting these off to whichever, internal regulatory agencies or just for internal meetings. Really, really nice tool that we employ. And again, same functions hold true. You're still able to annotate or reject the wells based off of what the software is classifying. And then it also has the sensitivity to detect cells that are in fact just on the point of division. So you can kind of see here, again, I apologize, it's rather small to see, but it will actually parse out and classify 2 cells that are in close proximity to each other as a doublet. And then ultimately, it would give it its rank as an overseeded well, which can then be further rejected by the end user. And it is also sensitive enough to, like I said, so because of the extensive training that we did, it's also able to pick up non-cell locations in the well here, indicative by this white box. So the scratches in the plastic, the debris, any sort of other non-cellular material will ultimately be picked up by the neural network in the automated evidence of clonality scan. Right. And then we've also employed, so I know it's not currently offered on the Cellmetric legacy software, but we actually do have the ability now to go through the Z-stack set of images on the Cellmetric X, further assuring the user that what they are seeing is in fact the cell. So you can kind of see as you thumb through the Z-stack of images there that the optical properties of the cell are in fact changing, further confirming the suspicions that it is in fact a cell and not a piece of debris or an artifact. It's a very high level overview of the neural networks and how they're used.